Okay, morning. So today we're actually going to discuss about this particular model. So once we make a protein structure, once we model a particular protein, what happens is that a particular protein we have to understand that whether generally any modeling software they gives us multiple structures. So suppose we went to a structure from either the iTeser or the Robita, they're going to give you 4 or 5 structures. Now, generally, the servers, the where you are modeling the protein, they have their own set of rules to tell you which protein structure is the best according to them. But it's always essential that we should actually validate that particular structure. Okay, and for this particular validation, there are several other tools that we can use. One of the tools that we're going to use today is the saves tools. Okay, this is actually given by the UCLA, and they host this particular server, and this server actually allows you to model that particular protein. So for example, currently we have this particular structure. Now let me open up another set of structures. Suppose we have this structure. Okay, so I'm on this particular structure. Suppose we have these 4 models, and so this is the model 1 that I'm opening up. And whenever you model a particular protein, it's obviously going to come with multiple structures. Now it's our idea to check which structure is better. Just a minute. This to model 2, model 3, model 4. So these 4 models are opening up. Now obviously the 4 models, as you can see, they are starkly different. All the structures are really different from one another, and that's why let it go. This is not the structure that we want. So obviously the first rule that I've already taught you, that you should always, now obviously if you take a look into this structure separately, they are not going to make any sense, right? They are not going to make any sense. So suppose you have this structure, the model 2, this is the model 3. Now the simple look can tell you that these particular models are not at all similar, but how are they different? So what can we do? You can simply going to go to the action button, and we're going to align that particular to the model 2, and then again click on that particular model 4, go to align, click on model 2 to align that particular model. The yellow line simply tells us how far that particular atom is from the original reference structure, and in this case we have taken the model 2 as a reference. So let me go. Yeah, so these are the 2 structures that we have aligned. Now the C alpha regions are generally aligned. So as we can see, the models are not exactly similar. So there are certain similarities that are inbuilt into them. You obviously can find out there are certain similar regions, but the mostly they are dissimilar. Okay. So in that respect, obviously we have to find out which of the model is better. Now, though it happens that they have their own set of rules to give you a better idea that which model is better, that they analyze, but sometimes it is essential to cross check that one. Why? Because sometimes it is not, sometimes the model that they say, that model 2 or 3 or the model 4, generally they give you the model according to the uh, decreasing order of their merit. So but sometimes the model 4, the list or the worst in case in respect to their ideology, sometimes that might be the best model. So for that we have to identify and analyze this particular structure, right? So for that we're going to use today this particular saves. Okay, the saves actually gives you the best idea about how your molecule behaves. Now one of the structure that I have already discussed and I have shown you, that we should always analyze the Ramachandran plot of each and every individual model, and that I have already shown you that you can do through the Swiss PDB viewer, right? Through Swiss PDB viewer you can obviously find out the Ramachandran plot, and you get to see which atoms are or which amino acids are not present in that ideal region and which are present in an unacceptable region. So and you can obviously minimize the structure to bring all those sequences on all those particular amino acids that are present in an unacceptable region to the acceptable version, right? So now what is this saves? This particular save is very easy to run. First what you have to do, you have to simply upload a particular protein model. So let me simply let you know, let's start upload a particular protein model. Particular thing that gets hanged. Don't worry, I already did that job, provided you that particular thing, but let me again show you. Just wait for a few minutes, it gets crashed. So first we have to do, we have to simply upload a particular model, any protein. So let me upload this model over here. Well, I'm uploading this particular protein. Mm, okay. Once you upload that particular protein, and then you have to simply click on run programs. So it's going to give you the list of all the programs that you can see, that you can actually run. So I've already done a job for you, so that because it requires some time. Now, first what it does, first it uses the erat function to gives you and analyze the non-bonded interaction. So first, what is erat? See, there are a variety of 6 molecules or models that and module that we can run to get analyze a particular protein structure. Okay, and among this particular protein structures, what do we have? The first is actually the erat. Okay, now here you see it analyzes the statistics of the non-bonded interaction between different atom types, and it plots the value of the error function versus the position of a 9 residue sliding window. And how does it do? See, first what the erat does is, so if you simply click on start, it's going to start the entire thing and it's going to give you the non-bonded interaction reciprocity. Okay, next you have the verify 3D. It actually does, it analyzes the entire model. Now obviously this is a 3D model that you have, but the 3D model came from a 1D structure, that's the sequence. So it analyzes the 1D sequence and it checks whether that particular 1D sequence can give rise to this particular 3D structure or not. And it analyzes some of the very important residual part like alpha, beta, loop, polar, non-polar, etc. And it compares the structure and the result for the good structures. Next, you have the prove. This prove program, what it does, it calculates the entire volume of each of the atom, and it simply thinks of atoms as a sphere, and it gives you a z-score, okay, for that particular value. And then there is a what check. Okay, what check. Now the thing is that this program, there are detail. If you simply scroll down, you get to see the detail of all this program. Okay, so who developed this particular program, what are the informations that are inbuilt into this particular program. See the what check documentation, the pro check documentation, the references, who developed, and we also get to see the Wikipedia page of some, right? Now, this what check actually analyzes the stereochemical property of your molecule. The stereochemical property of entire of your particular protein model. Yeah, it might be a protein model, it might be a DNA model, whatever structure that you submit. Now, generally people analyzes the protein structure because DNA double helix structures are mostly the similar, right? And finally, you have this particular project. Now, this particular analyzes the stereochemical quality of the protein, and what it does, it does by residue generation, or finds out the entire geometry of the particular structure and gives you the overall geometry, that whether the particular structure is right or not. Because sometimes what happens, the particular structure, they generally don't work in a very robust manner, right? They don't work very robust manner. Sometimes there are some places where the geometry is not obeying, because you have all the uh, what makes a protein molecule very stable, the entire geometry. Because if there is any unstable geometry in it, then obviously when you run the simulation, that particular part is going to get broken up, right? And that's why it is really important to see the Ramachandran plot too. Obviously the Ramachandran plot is not given by here, but you have to check the Ramachandran plot over there. Okay, so I've already run a particular job, and if you simply click this over here, if you start the job, it's going to get started. Click on the verify, it's going to start it. Click on prove, it's going to start. Click on this particular what check, is going to start, and pro check, it's going to start. Okay, now I've already done some analysis previously. Now, so see, first this gives you an error, gives you an overall quality. So with respect to 100%, my model stands at 85%. So it's a very good structure. I can't say it's a very good structure. It's 85% best structure. The quality is 85%. So obviously you can increase. That means there are certain areas that needs to be checked. There are certain areas that can actually be the geometry might be broken up, or there might be some steric hindrance, steric clashes that we have to look into. So these are the things that actually gives you the entire. Now, obviously, if you simply click on this result, it's going to give you the detailed result, the error result that you can get to see. Okay, so it's running. Let me click it over here. It's taking some time. Okay, uh, so let it run. Let me click it over here. Result. Okay. So now the verify actually, and as I've told you, it analyzes whether that particular 1D structure has been correctly modeled to the 3D structure or not, right? And whether each of the particular, suppose I have an amino acid that can have a alpha helix nature, or I have a stretch of amino acid that forms alpha helical structures, but the model somehow assigned a beta sheet structure to the particular thing. Now, obviously, these are going to give you a negative score. This is going to give you a negative score. So this is a very gross example, but they analyze like that and give you the entire thing. So obviously my is passing. What I get is that it passes. The entire thing it passes. So obviously there are 80% of amino acid that have that can pass through the entire region, and they have the very pretty good results. Okay. So now, so see in this particular case, so I have already started the ERAT score, the detailed ERAT job. So this gives you the entire overall score. So this is the overall score. So what it does, this is the error value that it says the how much time it takes and all this entire thing. So this is just a general idea about how your protein structure is. But you can obviously download this also, because sometimes you require to show your structure that you have already modeled this structure. And I'm selecting this model due to this analysis. Now what you have to do is, suppose if you have these 4 structures, you have to run all the 4 structures. If you have 4 structures, then suppose in my case I have these 3 structures, so you have to run these 3 structures one by one, and you have to see whether which model has better structure, which model gives the overall quality factor better score, and you have to give the proof it like that. So you can either download it using a in the form of PDF file, or you can simply download as a PostScript file, right? So you can simply click over here and it gets saved into a system that you can actually analyze and send or show someone. Similarly, this is the verify 3D. So this is the PostScript file that have been downloaded. So in the Adobe Photoshop, yes, but it's better that you download in PDF if you're not well versed in PostScript files. Okay. So next it shows you the verified result. Okay, the entire verified result. So what it does, it gives you that yes. So see, this is the basic amino acid that is given there. The entire amino acid. Okay, amino sequence that it gives there. Okay, so this entire sequence gives you the average score. So these are the raw scores, that's green, and this is the average score of the entire protein. And if you simply hover your pointer, you get to see how much your particular molecule is faring up, where your molecule is faring up according to the verified 3D and all. Okay, now obviously it this particular you can again save this particular molecule. You can simply save this, so you have the save data to file. You can save it as image. Okay, so since this is the basic line, this is the main basic line on how far the molecule is behaving away from that particular score, and that gives you the entire result. Okay, so after the verify 3D, I am going to get to proof to get the detailed job. So what does proof get? The proof simply gives you the idea that how many models or how many protein atoms are that are there that are present in the outlier, that you can obviously find out from the Ramachandran plot itself, right? So in this particular case, you see there are buried protein outliers from the first model. There are 4% of proteins that are present in the outliers. Okay, so how to get to see that? So see, this is the entire score. This is the entire score. So what it say? It says that how much model or how much molecule. So this is not showing the entire thing. So let me download it. So this is a PDF. Okay. This will download a PDF. Some of the parts is not being seen. So yeah, you can see here. So it gives you the score, how much more the average z score, the z score RMSD value, and obviously the distribution of the atomic z scores. Okay, so how many percentage of the proteins are in the outliers. So if there are more proteins than outliers, it's obviously better that you have to run a simulation, or you have to minimize the protein. And if you even if you don't run the simulation, you can minimize the protein using that particular that I've already shown you, right? That Swiss PDF viewer. In Swiss PDF viewer, you can minimize the protein, and it requires many simulation steps, many minimization steps. It might require almost 50, 20 to 30 minimization steps, and that can actually bring in all those outliers inside that particular in the in that particular zone with the acceptable zones. Okay, and this actually gives you the entire the residue by residue z score of the entire thing. So this is the maximum b factor. I mean, I have already discussed the b factor, right? That means how much your model is going to fluctuate, how much a particular atom or a particular residue fluctuates from the original position. So if the B factor is high, that means it is a very flexible molecule. And obviously a very flexible molecule with the very flexible residue, they're going to have high B factor. And high B factor means that particular stretch might not be very minimized. It might show random steric hindrances, right? So that is the thing. This is a total structure based. So this, you can see this particular triangle shaped these cubes discuss that these particular residues are not scored, whereas this particular box indicates that yes, this is the score. I can see the accessibility obviously. So the molecules that are present in the outline, the present in the polar molecules, they have high accessibility to that to any other molecules of water, whereas the molecules or residues that are buried inside, they have very lower accessibility, right? Because they are not going to get inside. So you get to see the entire protein, and they divide it according to the backbone and the side chain as well. So obviously you can simply see, and again you get to see the entire detailed chain analysis obviously. And this gives you an entire nature that how much it is varying, how much better your structure is, which part is helix, which part is strand, which part is, and what is the random coil structure. So this gives you a pretty good analysis of the entire structure. Okay, so this is the save proof. Now this is the what check. The results have been completed. So just a minute. See here what it does, it simply tells you the idea that whatever this particular molecule they are actually showing you or not, right? It gives you that particular idea. Just a minute. Yeah. So see what it does. It's going to take some time. This red indicates that they are not very good, so it must be taken care of. Okay, and the stereochemical parameters are not up to the mark. So what are the errors that are there? So see this particular first model, it has missing cell uniform. There is no scale matrices given in the PDF file. That's why it is showing the red. So it's get good. Okay, I get to what to know about that particular thing. Again, you can get to see, you can click all the different amino acids that are there, and there are the different amino acids. What is that entire check? So they're either saying the not crystal card is present or not present. So these are the the issues that are there. Now generally the crystal card issues and these card issues are not required. These are not generally required. Okay, they are not required for your analysis. So that particular what check you don't have to check. The major is the particular proof and the pro check. Okay. So it also gives you the entire idea that how much your entire model is. So it checks the entire protein. So it has done 8 evaluations, and among which there are 4 errors, 1 warning, and in the 3 parameters the molecule has passed. Okay. So what is the warning? So first you get to see the summary. If you simply click on the summary, you get to see the summary. So the summary tells you that this is the entire safe job. So how many? So there are actually 4 residues that are among the 190 residues. There are 4 residues that are not present, that are present as the outlier. There are several parameters that are, see, in the side chain parameters 5 are better, 0 are inside, 0 are out. So that means it's better. Very good. In the chi1 chi2 plot, you get to see the 4 level residues that are present outside. The entire G factor, the dihedral constant, the planar group, all these are given further. Okay. But the thing is that this plus charge, what is the plus? So that gives you the plus means it's a warning, and the star means there are error in the particular summary. Okay, and you have to check and you have to investigate further. That's what they are telling you. See this chi-square plot, the residue property, we have to check it. So first it gives you the error, the Ramachandran plot. This is the Ramachandran plot. It's going to divide you according to the different amino acids to give you how much they are faring. So here see alanine, the 522nd alanine is present in the red region. So that means this particular molecule is present in the outlier. So you have to check it. Now this particular things can be done if you simply minimize the structure. Once you model a structure, after the modeling, and obviously you have to analyze the entire thing, and after analysis you get to see that which model is better. Then obviously first don't look into this particular thing. If the model is very better, good, or else you need to minimize the structure. It is obviously better that if you minimize all the models. If you minimize all the models, then you are going to let go of the Ramachandran plot errors generally, because they are brought into the, now in fact you know that minimization also uh, removes the steric clashes. There are several steric clashes that the instrument simply removes, or the job simply removes, and that gives you a pretty good idea that yes, if the particular thing is removing the what should I say, if they are removing that particular errors, then obviously it's much better that you get to see that particular option over there, right? So that is very good option, that you can get to see. And obviously really the Ramachandran errors are simply removed by minimization, but it requires, you can't do a single minimization, it requires many single minimization. Okay, In the chi-square plot, you get to see that all are in the okay. It has passed the main chain parameters. You get to see it. The molecule is inside this particular, obviously. So it is, everything is better. And in the end, you get to see the plot statistics. There are omega angle deviation, the bad contacts, the zeta angle deviation, the hydrogen bond energy standard deviation, the overall G factor, the number of data plots, the parameter value. You get to see the entire panel, the stereochemical parameters. Okay, every parameters are given there. It's a very self-explanatory thing that you get to see. Okay, so in the sidechain parameters there are some errors, obviously there are errors, and you get to see the errors obviously, but obviously as I've told you that please perform the minimization, because that ought to release and remove all these, generally all the errors that are there. So for the residue properties and all, some it is showing you that it is passing the bond length, angular passing, the chirality factor. So obviously they are all passing, so it's a better structure. It is a very good score. It's good. Now obviously you have to understand and you have to give the entire thing. Read. You have to read the entire thing, and you can obviously download all the file in PDF file so that you can save them, and so that you can submit and let them know that what are the things that are issues, why I have selected this model, so it is showing this better structures and all. So even after this particular, whatever you do, after you particular get a job, then obviously make sure you analyze the structure, the model that you select, you analyze that particular model, because without analysis you can't for sure say that which structure is better. But obviously if you work a lot in these particular models and all, it is bound to give you the idea that this particular structure is better, that particular structure is better, and simply taking a look into a protein structure, you might feel that this structure might be better than the other. But obviously for scientific publications and presentations, you require to have hard proof, and this actually gives you a pretty good proof that how much or how these molecules and they all behave and all. Okay, so that is the issue, and that is the thing that you can do and check. And so this is in entirely the everything that you get to do, and while you perform the analysis, while you perform the protein structure modeling and everything. Now the last I'm going to give you one more thing. Suppose let's go to the particular file. Now whenever you perform the model analysis in ITESA, what they do is, I'm going to find out many models, right? So the model that you have, the best model, for the best model, they give you the, since it's a homology model structure, they give you the file depending upon which the homology model structure have been analyzed. For example, check here, this particular model 1, it has been developed because of this particular PDB files. These are the various models of the PDB files that have been used for homology modeling. Okay, but how would I know that this particular files that they have selected, these are the very good structures? Because if the reference structure is not good for homology modeling, then the entire structure might not be good, right? We have to check whether the homology modeling is good or right. So we have to check, and we have to know that if the homology model structure is good enough, and for that you have to check the reference structure. So how to check the reference structure? That's a very good question, and you have to see whether the reference structures are good enough or not. So for that we have to perform sequence alignment. Sequence alignment, because they develop the homology modeling according to the sequence alignment, and each algorithm, each server, each program, they have their own sequence alignment algorithm, depending upon which they align the PDB files found in the RCS PDB and gives you the better model structure. But it's always better to make a cross check. So for that, how can you do? We are going to use this cluster W. We're going to use cluster W to make sure that all the sequences and alignments are better. So what to do? Suppose you have to simply let find the structures that are used for your homology building model. You can get to see the structures that are there. You have to analyze, find out the sequence, and you have to simply submit the sequence over there. So in my case, just a minute, I actually have a structure. So I've already made a list of few molecules. Okay. So I'm not selecting all the proteins. I'm only selecting a handful of proteins to give you the idea. Okay. Let me select every. So suppose this is the protein that I've selected, and after that what you have to do is you have to simply upload that particular sequence over here. How to upload? Now first, obviously, what to do, we are going to select the output format as cluster, and then you have to perform the pairwise alignment. Either you can give slow. Now it is always advisable to give a slow because it is very slow and it gives a very accurate result. But for this class, I'm going to use the fast approximate method, and then you have to simply upload the faster sequence. That means the sequence that you have get together. Now obviously you go to RCC PDB website and you get to see the structures, and you can simply submit the structures. Okay, and once you submit the structures, you get to see that. So I have uploaded the faster format, all the faster format in the single go. Okay, and after you upload the faster format, what you have to do, you have to simply click on execute multiple alignment. That's all. Now it's going to analyze all the structure sequence and give the result. Now obviously I have uploaded here 11 sequences. This is a hexokinase gene that I've selected. That is for human, rat, mouse, yeast, bovine. Ponab is the bonobo chimpanzee. Then a Solanum tuberosum, that is the potato. The absparch, that is the seahorse. Then not seahorse. I forgot the name of this particular species. This is tobacco. Felicia is the cat, and this is the orangutan. So first it gives you the which sequence that you have uploaded and what's the sequence length. So 970, 980, 974, 485, like that. And now it's going to perform a sequence pairwise alignment. So it's going to align 1:2. So to align human and rat, and they're going to say how much similarity we have. So see, human and rat have 92% similarity. 1:3 means human and mouse. Human and mouse have 90% similarity. That means rats are much more closer to us rather than the mouse. Okay, rats are much more closer to us. And human and yeast is going to be very low because we are different species. We are not a single species. I mean, not a single species. We don't belong to the same class of don't belong to the same class, right? That's a fungi. Okay, so you get to see the different form over here. Then obviously like that 1:3, 1:4, 1:10, and all that. It's going to analyze every pair with one another and give you the entire result. But if you come down, then it's going to give you the how much the entire sequence is there. Now the Felicia, as you can see, the Felicia or the cat has a cat hexokinase is very much long, 1056, whereas the average structure is 918, 917. So 920 is the average structure, whereas 1056 the cat. That means cat has some added mutation. It has some additional mutations, insertional mutation that is present in the cat, right? So once it analyzes that everything's together, so here we can find out that yes, every molecule is more or less same. YDP, TPN, VH, GGG, everything is more or less same. That means the same. This particular domain is highly stable. This particular domain is highly stable. Okay, so once you upload this particular molecule, since I've already uploaded this cat, you simply remove the cat. If you simply remove the cat, you're going to get a change in the sequence analysis. That is rat, mouse, yeast, bovine, bonobo, chimpanzee. Wait a minute. Solanum tuberosum, tobacco. Yeah, Felicia. Let me remove Felicia. Now let me execute it. So now more of everyone has more or less same structure. Okay. Yes, obviously it's going to be less because I have this 485, so it is really less the yeast. So see, once I've removed that particular thing, you can get to see that yes, the rest of the part is really the same. The orangutan, apart from the yeast, the Solanum tuberosum and that particular thing, uh, everything is more or less same. The sequence is more or less same. So this is what the sequence homology is. So once you upload your protein, you have to see whether the sequence is similar or not. Because if they are homolog structures, they ought to have some similarity, right? And if they don't have proper homology, there are going to be a lot of differences. Now, obviously, you're going to come across several identities. What are those? Now, see, the star indicates that this particular sequence or this particular amino acid is similar in every species. Now you might say that here also is everything same, but why don't we have star? Because the East Solanum tuberosum has few. They don't have that particular amino acid. That's why can't get to see the star over here. But here all the amino acid in every species that is same. So that's why you get to see a star. You get to see a double colon. What is this double colon? The double colon gives you the idea that these are similar amino acid. Now we know that amino acids are more or less similar. First we have amino acid that are uh, there are few amino. We know that among all the different amino acids, they are divided into various classes. There are hydrophobic amino acid, hydrophilic amino acid, positively charged amino acid, negatively charged amino acid, right? Now, obviously, if you replace a positively charged amino acid with a positively charged amino acid, then it is not a very big difference. Approximately the length is same. See, the double colon indicates that this is almost similar. Okay, it's almost similar. Just give me a minute. Yes, sir. That means if there is a change from positive amino acid to a positive amino acid, that's not a big change. Because positive amino acid, even though there's a mutation in respect to protein, that is not going to change the protein structure or the protein function. Okay, suppose a protein at the particular domain has some hydrophilic nature, it's interacting with water. So if it gets replaced with another hydrophilic amino acid, it is not going to replace or reduce the protein function. It might substitute it. Yes, the hydrophilicity might be little less or little more, but it's going to be similar, right? So that means it's simply like a prosthetic. If someone loses an arm, and suppose someone, it's simply like an surgery. Suppose you have your people are getting kidney transplantation, right? So it is not your kidney, but it is working exactly fine. But you have to take some care because your kidney is going to be your own kidney was much more better than anyone else's kidney, but they give you similar result. So this double colon indicates that more or less similar, because you see everyone that is methionine, leucine, methionine, phenylalanine, methionine, isoleucine, all are similar kind of amino acid. And the single colon, the single dot gives that they are similar but to a lesser extent. They're almost similar. Which means they're similar but almost similar. So you see asparagine, serine, aspartic acid, asparagine, and then you have glutamine. So not exactly similar, because there is some, you see aspartic acid over here, right? And you see glutamine over here. Now obviously aspartic acid and glutamine are not exactly the same, because glutamine is NH2 positively charged, whereas aspartic acid is negatively charged. So that means there are some charge differences, but rest of them has almost similar. That's why you get to see a single colon. And the one that we don't have the single colon, that means the structure has entirely changed. The properties absolutely changed. Okay. So the more number of stars, more number of double colon indicates that more conserved that particular domain is, more conserved. So what you have to do, you have to upload. So once they model the protein, since they're taking homologous protein, it's not that they are going to have 100% similarity, but they ought to have at least 80% similarity, 70% similarity, 90% similarity. And if the similarity lags below that, then obviously it's not going to have a very good structure. So that is why the sequence alignment is done, because if you see that with some of the models, your with the original structure, reference structure is suppose 1, and with the reference structure other models are not going with that part, I have 98%, 92%, but if there is 28%, that means there is no similarity at all, then this is the model that you can't use. Then you have to simply reject that model. So obviously then you have to rerun the entire modeling, rejecting that particular PDB ID. Or if you have any PDB ID in your mind that you get to know of, then you have to incorporate that particular PDB ID, right? So you have to do this for your better structure. Okay, so this is another way of cross verifying that your model structure is highly good. It's very good. Clear? So this is always better to structure your model. So that is how you get to have a very good protein structure that you can analyze, and obviously doing all this is going to give you an absolute protein structure that you can move forward in your research. So there is no one can question your intent. No one can question that if it's whether your sequencing modeling structure is good or bad, because you have absolute every proof that yes, you have cross-checked them, and obviously. But obviously, one thing is very essential, that whenever you model the protein, make sure you run a MD simulation for your protein. You run an MD simulation, because if you don't run an MD simulation, it's not going to hold. Okay, so running an MD simulation is absolutely necessary for your protein structure. Yeah, so keep that in mind. Okay, okay. So we are concluding our class over here. Okay, so thank you.